All right, and good morning, everyone in the audience. It's great to be back here all together again and touching base again on how far we've come over the last year. I am fortunate enough to be joined up here on stage by Stewart Abbot, who's the Chief Development Officer at Fate Therapeutics.
Good morning.
Good morning. As well as David Chang, the Chief Medical Officer and Executive Vice President of Research and Development from Kite Pharma.
Well, thanks for having me here.
Tom Farrell, the President and CEO of Bellicum Pharmaceuticals.
Good morning.
And last but certainly not least, Christian Homsy, the CEO of Celyad.
Morning.
All right, well, as you can guess based on the topic, the first thing we want to talk about this morning is progress. So I'll open up my first question. Maybe, David, I'll direct first towards you. If you think back across the last 12 to 18 months, what are the areas where you think we've made the most progress? Where have we really moved forward?
Well, great question. So I'm going to focus more in terms of cellular immuno-oncology. I mean, that's sort of my area of specialties. I think, there has been a lot of promise of the cell-based therapy in the space, and the promise is really, fundamentally changing the outcome of the patients with cancer. So when you think about the history of, oncology drug development, it's always been multi-drug, chronic treatment and treatment that, provides incremental benefit, that usually is measured by the increase in the median overall survival, without really affecting, long-term outlook of the patients. What the cell therapy has done is, lifting the plateau of the Kaplan-Meier curve, where some patients go on for many years without recurrence of disease, or sometimes without any evidence of disease. I think that's where the T-cell-based therapy, along with other cell-based therapies, are making headways. To me, I think, sort of speaking for my company's experience, translating a single institution experience into a multi-center clinical trial, I think, which is taking one step closer to commercializing cell product. I think that, we are doing it, others in our space are also doing it, and everyone's moving along with some glitches here and there, but overall, everyone's making a positive headway. And in terms of, we recently at Kite Pharma announced, outcome of the pivotal data, which essentially reproduced what others from single institution studies at NCI or UPenn or Fred Hutch have been talking about. So I think that was really reassuring that the cell therapy immuno-oncology is here to stay. And now, extending that into, in other spaces, I'm also very excited about, what's going on in the neoantigen space, especially in solid tumors. I think it's relatively early, but some of the data that's coming out of that space is really tantalizing and really makes you think beyond the heme malignancies and also into the solid tumors.
Great comment, and we're going to come back to that in a few minutes. Maybe I'll ask Christian if you'd like to comment. Where do you think we've made a lot of progress? What makes you optimistic about our moving forward in this field?
Well, I think to David's point, indeed, it is, since now probably 2, 3, 4 years, we've been seeing data that has really moved the needle. We're seeing things where there's a real impact on patient lives, both in terms of overall survival as well as in terms of quality of life. And I think also that companies are now starting to work on the manufacturing part of things, because it is absolutely critical. I mean, we can have great products if we cannot get them to the patients. We don't have a product, so we absolutely need to work on that very hard. And I think that's basically where the field has moved over the last couple of years, and that's very encouraging.
So as you all think about what kind of progress we'll see in the next 2 to 3 years, maybe Stewart, I'll start with you. When we come back here again in a couple of years, what do you think will be the most different? What type of progress do you think we'll see?
I think there's a range of therapies that are on the cusp of sort of being real. David highlighted, his company has extended the experience from a single center into multi-centers. They're in phase 2. There are other groups, Juno, Novartis, that are in phase 2 studies with engineered T cells. Those studies are moving spectacularly quickly. There have been some setbacks along the way, but those studies are moving very quickly. So I think if we look, 2 years, potentially 3 years out, we should see some approvals in this space with cell-based therapies. I would be surprised if we didn't. I think that possibly a topic that we'll come back to, but spectacular rise of checkpoint inhibitors in the immuno-oncology space, and when we think about cell-based therapies, we're probably going to have to think about cell-based therapy combinations. And another 2 years down the line, we may see some spectacular, some really good progress when understanding the best combinations. So I think that's a couple of things that we'll see in the future, very positive things. There'll be some setbacks along the way, but those are the very positive things that we should see.
Excellent. Tom, your thoughts?
Yeah, so I think we're already seeing the data move beyond CD19. We're seeing some other targets like BCMA, MYESO1 that are looking quite encouraging, but we don't have a lot yet to put our hands on in the solid tumor setting. So speaking for Bellicum, we have a couple of programs now going into the clinic with controllable T cells. And within the next couple of years, we would hope to have meaningful data from those programs in the solid tumor setting. And I think that's sort of the point at which I think this field really becomes mainstream. We're hoping as well at Bellicum to have a approved product in that time frame. It's in the setting of stem cell transplantation, which of course is often overlooked as being a form of cellular immunotherapy that is here today. Approximately 25,000 stem cell transplants, allogeneic stem cell transplants, are done every year in Europe and the US. And essentially what we're doing is we're taking kind of the current standard of care, which requires a full match between the donor and the patient, and we're saying, can we make a half-match transplant safe enough? Everyone has a half-match donor available to them. And the data that we presented to date is really very encouraging that we can achieve curative outcomes in these patients with very acceptable toxicity profiles. So we're excited about that. We hope to have it approved and on the market, certainly in Europe, within the next 2 to 3 years. And we're waiting on end of phase 2 discussions to see what the pathway looks like in the US.
Excellent. Stewart, you commented on combination therapies, some excitement around maybe combining with checkpoint inhibitors. David, maybe I'll ask you to comment. What do you think of as the potential for combining therapies? Which therapies do you foresee having a significant impact on patient treatment?
So in this space, in the immuno-oncology, I think when you look at it, you can come up with a rationale for combining, various different pathway inhibitors. And certainly there are products that are approved in that setting. My view on the combination is let's work with something that has proven efficacy and combining with something else. For us, we have previously commented at Kite that we will be starting a combination study of our lead program, KT-C19, CD19 CAR, together with PD-L1 antibody. Based on scientific rationale that includes upregulation of PD-L1 in lymphomas, and also the fact PD-1 is upregulated during the T-cell activation process. So, that kind of combination, will continue. But I think, also, when I look at this space a few years down the line, the beauty of cell-based therapy is you using the cells as the tools to apply synthetic biology into stepwise altering the behavior of the T cells to do exactly what you want it to do. So, take a step back. What is the chimeric antigen receptor doing? 2 things. It's redirecting the T cells using the single chain variable fragment as more or less like a GPS. And also it's linked to the signaling domains to activate and allow the T cells to expand and kill the targets more effectively. So Now, when you move to combination, from that single step of engineering, you can add on other steps of engineering, either using gene editing to knock out PD-1 or other checkpoint inhibitors that are part of the T cells, or potentially providing other agonistic signals to the cells to have the cells expand more effectively. So there are many different combinations, and I think certainly the initial proof of concept will come from doing a standard, more traditional combination therapy, but once that proof of concept is established, I think the path is very clear. Through gene engineering, you can combine and package everything into the cells.
Excellent. Before we shift topics, Tom, Christian, anything you'd want to add to that?
No, I think it's early days as yet, and I agree completely with David that it makes logical sense to combine your cell therapy with a checkpoint inhibitor first, because you may not have the right one, right? You don't want to engineer the T cell until you know you have the right pathway. But yeah, I think that's coming is going to be important. The role of gene engineering or gene editing more broadly, I think, is still somewhat up for debate. Whether you can truly use gene editing to make, for example, an off-the-shelf allogeneic T cell, I think remains to be fully demonstrated. But in terms of engineering out important pathways, I agree.
And
also, if I can just add on to this, I mean, in this space, in the cell therapy space, I mean, when we think about combination, we should think about not just in combination trials in patients, but also using small molecules or others or cytokines during the manufacturing, ex vivo manufacturing process to make the cells, or let the cells differentiate or kept in cells in certain phenotypes. And I think that's probably something that's much easier to do. I mean, that's a manufacturing process, and certainly we are doing it, and others, I think even Fate is heavily into this space. And I think that probably is another dimension of combination that we should be thinking about in this space.
excellent.
I think just to pick up on that last point, I mean, the ability to modulate cells, the ability to guide cells will certainly increase in prevalence over the next couple of years. But I think also that we're now learning from the appropriate species. We're actually in clinical studies in man, and so those translational studies, understanding what happens to cells after they actually go into these patients, are absolutely critical with regard to helping us understand how to guide, whether to guide through genetic engineering, whether to guide through small molecule programming. We have to be in the relevant species to do that, and we now are. And so the learnings that are coming out now and over the next year or two, I think, will allow us to logically modulate cells.
Excellent, thank you. So as we look toward the future, there was a comment made earlier about the use of these types of therapies moving towards solid-based tumors. Maybe, Christian, I'll start with you. What do you think? How applicable will these technologies be to solid-based tumors going forward?
Well, I think solid tumors is a very different beast, and it will require an approach that builds on what has currently been done and pioneered by all our peers, but also adds to it elements that are going to make it more effective than today it is. I mean, the challenges are known. How do you get into the tumor with the cells, with the microenvironment of the tumor? How do you deprive that tumor from its blood supply? How do you make sure you do not target on target of tumor toxicity? How do you kind of create an adaptive mechanism so that you actually teach the cells that you haven't taken out to actually act against the tumor as well? So all those things are programs that we will be soon starting 7 different tumors before the end of this year, of which 5 will be solid, and addressing all those different aspects. Now, I'm not sure if we have the recipe, but certainly we're trying to target each one of those paths, because I think just a cytotoxic activity which seems to be to a great extent working greatly in liquid tumors, may not be enough in solid tumors.
Yep. Anyone like to add on to that?
I think I'd sort of concur with the points. If you'd asked me the question 5 years ago, I probably would have answered just with one word, and that's microenvironment, which was highlighted. If you ask me the question now, it's probably 2 words. It's microenvironment and neoantigens. So we have to understand, further understand the microenvironment of the solid tumors. Not all solid tumors are born equal. And understand the antigenic profile of those solid tumors such that we can appropriately target. So if we more fully understand neoantigen expression and more fully understand microenvironment modulation, I think we'll move into the solid tumor space. And people say that this is, some people would say this is insurmountable. I don't think it is, but it is a significant challenge beyond the liquid tumors for cell-based therapies.
Based on the concept that the microenvironment plays such a strong role, can we apply what we learn about one solid tumor easily to another?
With limits, yes. I think yes. I mean, David's probably a better place to answer this from a medical perspective, but yes. I mean, I think there are certain learnings that we can apply broadly, but there are some features of different solid tumors that are unique to those tumors, whether it be the location in the body, potentially behind the blood-brain barrier or not, whether it be the development status of that tumor. There are differences, but we can apply some of the learnings, yes.
Excellent.
I guess I'd just like to, so what wasn't touched on, I think there are many factors. And maybe one question is whether, at what point is someone going to solve them all? But what we know about ALL is that you have tumor cells that are in the bone marrow, you have T cells that traffic to the bone marrow. There is no tumor microenvironment per se, and so the T cells proliferate. And of course, you have problems like cytokine release syndrome, which is where the T cells have excessively proliferated or resulted in the release of high levels of cytokines, which can, of course, be toxic. That doesn't happen in solid tumors, typically. Typically, in solid tumors, the T cells, it takes them too long to get to the tumor to encounter antigen. If they don't encounter antigen, then they don't proliferate and they don't expand. And oftentimes, they just don't survive long enough. So managing the cell population through the course of treatment, so getting the cells to survive long enough, they see antigen, then driving them to proliferate, so they proliferate reliably and consistently from patient to patient, keeping them around for long enough. So I don't know what Kite's current thinking is on how long you need the T cells around to have a complete response, but it's quite likely that in solid tumors you need them around a lot longer. And we see, for example, in the work that's been done with mesothelin, that typically whatever you do, 28 days the cells are gone. Okay, so how do you keep them around? And then what do you do with them after the effect has been achieved? Do you keep them around? Do you want them around for surveillance, or do you want to get rid of them to avoid any potential downstream toxicity? And that's where controllability comes in. So that's really what we're trying to focus on, is how do you control the cells to achieve this kinetic profile that you need to achieve in order to have a profound effect on the tumor.
Yeah, two challenges there: figuring out what the ideal profile is and then making it happen. Gene editing was commented on a little bit earlier. Obviously, innovation has played a key role in moving forward this field. David, maybe I'll start with you. What additional innovations do we need? What bottlenecks do you think need to be solved through innovations that maybe don't exist today or aren't fully applied yet in the field?
Wow, that's a very loaded question. I think, we have to sort of think about taking a step back. Even last 2 to 3 years, when I think about it, the new technologies that have entered into this space, the list is pretty long. I frequently say that, the whole field of cell therapy now has passed the critical mass, juncture. So it's continuing to expand, and it's really not an individual or individual company, working by itself, but really leveraging what others are doing and, understanding the new technology and applying it appropriately, for the need. I think that's going to be critical. Gene editing, to me, is one of the many enabling technologies that are out there. I think, other technologies such as providing, agonistic stimulus to the T cells. I mean, that's being talked about. Doing it in a cell-focused way rather than just providing, uncontrolled activation of the immune system. Packaging small molecules or other, cytokines onto the T cells to make it work more effectively. So I think there are many, new technologies out there. I would say that several are very attractive, but I can't really think about one that's really going to,
Can't
make
pick
a
a favorite. Any other comments from anyone else on the panel regarding innovation?
I guess I make just kind of the general comment. I think that I think we all recognize that the field has been transformed in the last few years, particularly through the extraordinary results seen with CD19 CAR T cells. Usually when I'm on a panel, it's with an investor audience, which is a little bit different. I'm a little hesitant to say too much that's technically minded with this group here. But I think what we've seen is a rush into the space. Huge amounts of capital are being deployed into this space. And a lot of that money is just kind of trying to chase the good ideas. And the first challenge is to identify which are the good ideas and which are really just kind of a repurposing of something else that really isn't that relevant. So I think sorting out the wheat from the chaff in the innovations that are currently occurring or that are real or not real, I think is part of the challenge that the space and its supporters and investors are working through still at this point. So I think, David has just touched on a few of the things that people are doing and working on. There's many, many more. And I think figuring out which of those are going to be important and which are not at this point is going to be quite difficult.
I mean, one thing I'd like to add, and this is a little bit selfish, but from a perspective of fate, but we look at gene editing of cells at the moment, and certainly, obviously, we've had successes with CD19 CAR Ts in the setting of patient-specific therapies. But I think as we go forward and we think about multi-gene editing of cells, it's going to be incredibly difficult to apply those very innovative, very useful editing technologies in the setting of patient-specific autologous therapies. And I may be running the risk of sort of alienating a lot of the audience that are working on patient-specific therapies. But imagine trying to cook dinner on a yacht in the middle of a storm. This is sort of analogous to multi-gene editing and patient-specific therapies because every donor is different, every patient is different. And so you have this wonderfully predictable molecular biology that you're throwing on top of these horribly unpredictable patient-specific cells. So one of the things that certainly we're working on and others are looking at, and David mentioned this with regard to allogeneic therapies and the like, and off-the-shelf therapies, is the ability to cook dinner on a level surface that's not moving. And what I mean by that is that the level surface is a continuous cell line, or it's a very, very reproducible cell type that you can then edit. And our approach is obviously to look at induced pluripotent stem cell-derived NK cells and T cells as that incredibly stable platform, which you can then apply editing on top of in a predictable fashion. In the absence of that ability to predict, I think we're going to take an awfully long time in developing these therapies in autologous settings.
Yep, that's an excellent point. Let's talk a little bit about the topic of commercialization. A few of you highlighted that it's quite a feat to move from a single center trial to a multi-site trial. How do you move from being really affected with 10 to 20 patients to getting to the point where you're treating 100, 200, 1,000 patients? What are thousands and thousands of patients? How long do you think it'll take us to get there? And if you could talk a little bit about how you think that would evolve. And I'll open this one up to the whole panel, whoever wants to jump in first.
Well, Celyad has gone that way. Our first product is a cell therapy product for heart failure, in which we moved from phase 2 to phase 3, and we've treated north of 300 patients now across the globe with that. And it is a very, very complex endeavor. And it has to be tackled right from the start of development, I think, because indeed moving from quality control site level manufacturing to a much broader place, even if you centralize the manufacturing process, is extraordinarily complex. So we as an industry have a duty to actually solve that, because it's not going to be solved by traditional methods. I don't think it's going to be solved by traditional methods. I think the approach of allogeneic is, or cell lines, is one that also has its own challenges. And of course, there are pioneers in the field that are trying to address those. But there's many ways to skin a cat. I mean, you can do this in a very different ways. And I think we have to be more creative, because frankly speaking, if we're not, this field will never take off.
And maybe just a follow-up on that. To do that, have you had to engage partners or others outside of Celyad in order to make that a reality?
Initially, no, because, it depends what you call partners. If we're talking about big pharma or large biotech, no, because they were very, and still are very skeptical about the approach, the business model part of the approach. Maybe not the science, but at least the business model part of the approach. And rightfully so. We haven't yet proven our capability to actually deliver return for our investors with these products. So they have to be. However, there are companies out there, and we've partnered with Invitech, and there are others out there that can help you into that process, because you can't have all the skills internally, of course, right? Yeah. There are people that are much better than us at doing those things, and we just need to partner together and make it happen.
Excellent. Tom?
Well, I guess I would argue that we're a little closer than the question might suggest. If you look at Provenge, for example, I mean, it's kind of the object lesson in how not to do it, supposedly, but that product is being delivered to patients today. I believe, thousands a year, and each patient is getting 3 doses, and it's a fresh product. So I think a key innovation that everyone, born after 2000 as a company has the benefit of is cryopreservation. So with products that we're shipping today to multiple sites, multiple countries, we have post-thaw product viability that's a median north of 90%. So, it used to be, and when Provenge was developed, that cryopreservation, I'm sure there are people in the audience that have that technology, that wasn't available when Provenge was being developed. So just the fact that you can freeze the final product allows you much greater flexibility in managing the logistics. Okay, provided you have time to get it to the patient, then you can use that cryopreservation to manage your manufacturing, scheduling, to avoid the issues of loss of potency and activity, while the product's being made or as soon as it's shipped, etc. So I think we already have an example of a situation where, while it's not, probably the ultimate, I think we've seen someone be somewhat successful with a product that is really very much more challenging than the ones that I think we're all developing today.
True. Anyone else want to comment on that?
When I think about the cell therapy space, for those, some of you have been in this space for a long time, when the, monoclonal antibodies were coming up, the biggest question that everybody was asking was, how can you manufacture the antibody to treat, so many people? And nowadays, if you ask that question to companies that are making monoclonal antibody, they'll say, which planet are you coming from? So to commercialize, I think, to me, one, you got to have the legs. You got to have the efficacy, the benefit, that you can offer to the patients. I think that's pretty critical. And CD19 CAR has been a tremendous sort of positive element in this space. Being able to generate objective response in upwards 3/4 or sometimes going up to 90% of the patients depending on the indication. That is a really good starting place. And as I talked about, translating that single institution experience to multi-center, I think that's critical. And then, being able to handle the manufacturing and the cell logistics. If you're dealing with the patients who are dealing with life and death situation, manufacturing of the cells is not something that you can just think about as, well, we'll manufacture. It has to work almost 99% of the time. And, when we started our clinical study, we had a lot of questions about how consistently we can manufacture the cells for every patient. So far in our clinical programs, we treated over 100 patients, and we have had very, minimal, treatment manufacturing failures. And also, while that's going on, all the backup processes are being built into the manufacturing so that, one day we can get to, almost every patient, every time type of situation. And also, manufacturing, I think, the streamlined process and working with others in this space. I mean, we are not the only one who's manufacturing. Certainly, we learned a lot from Dendreon experience, but others, GE Healthcare, GE Global Research, and there are other players in this space with a lot of expertise in the manufacturing. And trying to leverage that, I think, that's going to be, one of the critical elements to make the commercialization of cell product, a much faster process than what one could do as a single company. And certainly, Thermo Fisher is a big player in this space that we also work with.
Thank you, David.
And so, to me, sitting here, I mean, I think 2 years ago I would say, well, it's probably 50-50 cell therapy, CAR T would be successful. Right now, 2 years later, I think most people would believe that it's probably 80-90%. And as Tom had sort of alluded, that's just the beginning. Especially even if you don't go into the solid tumors, in heme malignancies, if you can cover B-cell malignancies, multiple myeloma, and AML, you are addressing collectively 3 diseases that incidence in the U.S. alone would be about 150,000 new cases a year, where more than 50,000 patients will die from the disease themselves. Those 3 indications. So if you can just work in the heme malignancies where proof of concept are more readily available, you're making a tremendous, providing tremendous benefit to the entire healthcare system. Not to mention all the patients who's looking at very grim outlook in their lives. So to me, this challenge about commercializing, I think that's already being addressed pretty to the fullest. And the challenge now is going from a single indication, single product to the multiple products to really let the field grow.
And what did you want to?
I'll just comment on a couple of things. So, I mean, going from 10 patients to 10,000 patients kind of sounds sort of insurmountable, but Thomas highlighted this earlier. It's actually already been done. We transplant 20,000 plus patients every year, hematopoietic stem cell transplant. That system is managed largely in the US by the National Marrow Donor Program. Speaking to somebody from the NMTP the other day, and they highlighted that even on 9/11, they managed to get their products around the US, 2 patients within 48 hours. Now this was, the US was a no-fly zone on the 5 days after that time, and the NMTP still managed to get their products to patients. It's a logistical challenge, and there are, going from 10 patients to 10,000 patients, there are huge logistical challenges with regard to manufacturing, the cold chain distribution, whether it be cryopreserved or high temperature. They will be solved more broadly, but we have models to look at today where transplant products can be produced. We can enhance transplant products, but Thomas and our company are working on that. and we believe it's feasible to actually get these products to a vast number of patients. We'll probably touch on it later, but beyond the boring bits of the logistics, we've got some challenges ahead of us with regard to reimbursement. So, that's another part of just making this real for, many patients as opposed to tens and hundreds.
Yeah, it's a good point, and I know a sensitive topic in the industry in terms of pricing and reimbursement and topics such as that. A few of you have commented on technologies that would help us make a better cell. Tom, I think you commented that you have a post-thaw recovery of 90%. So have we solved the dosage problem? Is our real focus now need to be on making the cells that are in there more effective or different in some way? Or do you think, and I'll open this up to anyone on the panel, do we still have challenges to get the right number of cells to the patient?
Yes.
I think that, so I think that's the wrong question. The question is getting the right threshold level of cells in the patient. So regardless of how many cells you administer, do they expand sufficiently, right, to have an effect? So I know David probably has a better perspective on sort of dose response at this point, but I think that's more the question. It's not how many you give, but how many, what is the peak level that you achieve? Because you need that peak level in order to have an effect.
Yeah.
I was reading a paper this weekend on which was published a bit earlier on BCMA CARs and the work from NCI. You're probably very familiar with, David. And it's interesting to see that some patients, to your point, some patients will react to a certain number of cells injected and others won't. And that is very closely correlated to the expansion in vivo. And so what is the model? Is the model of putting cells in, hoping they're going to expand, or manufacturing them in a certain way that they are going to expand, and then control them, which is what Thomas' company is doing very well. That's one approach. The other approach is, are we going more into a drug-like approach in which you are achieving a certain necessary number of cells injected, and then you re-inject a couple of 2, 3, 4 times, and to keep that kinetics over a certain period of time until you get to the treatment effect you have. And then the last complexity of that, the other complexity of that, what are you triggering inside the patient in terms of immune reaction that are not depending on the cells that you have injected, but there is a consequence of the cells you've injected. So it is a very, very complex multifactorial equation, I think, but we're at the beginning. I think we have a tool now that can do many of those things, and now we have to work on developing those.
Yeah, excellent point. So all of you have been in the field for a while. Obviously, the field has learned from sort of, I would say, the early pioneers. There may be in the audience people who are at sort of the beginning of that journey. Any advice you'd give to new startups on how to work through the challenges smoothly?
Smoothly?
Or perhaps how to survive the challenges when they come?
Well, it's a much different world today than it was 10 years ago when Bellicum started. So we had, in some respects, the luxury of time to refine the technology, to generate clinical data, to make, informed decisions about where to focus and where not to focus. I think today, as I mentioned earlier, there's so much money in this space. Things are moving so quickly that managing that as a startup has to be very challenging, and I would think very difficult to do alone. The other thing I would say that I think is probably apparent to everyone in this room is, don't underestimate the challenges, so particularly the challenges of manufacturing products, et cetera. It's not, you can't just, open up shop and start making product.
Any other comments? David?
Nothing that's earth-shattering. Only thing I would say is, when you start, obviously you have the technology platform that, is the foundation to what you're really starting with. Get the best people to work on it. I think you cannot underestimate what, the team and people can do and always surround yourself with somebody who's smarter than you and somebody who can really push and have the same vision of what the field will look like down the line.
I think in one word, focus. Especially for those groups and companies that are developing platform technologies, whether it be engineering technologies, whether it be different cell types. It's very tempting to go into scientific development plans and clinical development plans where you're applying technology broadly. And for small companies, they may not have the time, the money, the people to do that. So there's always a risk of missing opportunities and not going into particular therapeutic areas or indications. But there has to be, the advice is to have the discipline to focus on specific indications, specific areas, and to drive through into clinical development as quickly or as efficiently as possible. Not necessarily to dwell too long on the preclinical models and the mouse models and the like that might have a particular bias on that. I don't think they're particularly informative. To get safely into the relevant species, to learn from that clinical experience, and to focus on your clinical development plan.
And I'm going to ask my friends in the back there, is it possible to get a mic up and open it up for questions in the room? All right, excellent. If maybe we could just bring the lights up a little bit. We have a few minutes. We can probably just take a few questions from the room if there are additional topics that we didn't touch on that people have a passionate, burning desire to learn more from our panel about. Anybody? I'm sort of blinded up here, so if someone could help me see if there's any hands raised in the audience, that would be helpful.
This is a question about the pluripotent cell-derived cells. How easy is it to make those NK cells, and is there any concern about IPS not being as authentic as, say, the ES cells?
Surely. So actually, it turns out, none of this is easy. But relatively speaking, it's actually easier to derive NK phenotype from a pluripotent stem cell than it is to derive a bona fide T cell, CD4, CD8 population. So NK cells are relatively straightforward in that regard, and the purity of the population is good. Just going into the science a little bit, it's a little bit of an immature population, but there are many groups that are looking at the clinical efficacy of very, very similar cell populations, and we're working on further biasing the differentiation of NK cells and T cells with small molecules during manufacture. And we're having some, certainly preclinically, some great success with that. So I think we will see in the near future pluripotent-derived NKs, followed shortly by pluripotent-derived T cells.
Excellent. And we have a question in the back, I think, I see. Oh, one right here.
So I want to go back to Christian, one of the points that you raised in terms of sort of comparing, contrasting long-term durable X-fold expansion of cells versus the hit-and-run approach, where you come in with repeat administrations as a way of compensating for long-term durable expansion. I would kind of be curious to hear other panelists' views in terms of where that hit-and-run approach is and what data would be required to say whether that's a valid approach or not, or how are you guys thinking about that in your own internal programs?
Well, so maybe I'll take on that question. I mean, if I think about the history of drug development in oncology, I mean, and also I think this applies to, what happens in the, laboratory work as well. Chronic exposure to, inhibitor or anything, it always provides that selective pressure that will create something that will overcome the resistance. And I think that sort of underlies why the early pioneers in the treatment of leukemia, they went with a multi-drug combination to kill off all the cells from the beginning. So, that now when it comes to the days of immunotherapy, there's a little bit of sort of, conversation going on about that. Let's get rid of all the tumor cells and then, cure the patient versus let's control the disease through the immune surveillance. I have to say that I'm more towards on the, hit and run, if I borrow the phrase, to really eliminate the cancer. The chronic treatment, chronic surveillance, some vaccines do work, but in general, for the tumor treatment so far has not been that successful. There's always the emergence of resistance that will come up in that setting. And I think also, especially with the CD19 CAR space, there's a lot of questions about cell persistence and the durability of the response. Certainly, people in the field are measuring the cell persistence. I think that also has to change to not just measuring the cell persistence, but cell functionality. Just because you can detect the cells doesn't mean that they are functional. We learned from the checkpoint field that these cells can hang around, but are inhibited through the various regulatory mechanisms.
Thank you. You want to add, Christian?
Yeah, I think in your question you had an X-fold expansion. The question is that, one of the questions is, what is that X? Is that X similar in patient A, in patient B, in patient C, in patient D? And can you manage that? And I think that's one of the biggest challenge. And I think also having the same CD19 CAR present for a long period of time, you also see CD19 shedding and escape mechanism, and this is how patients relapse. So you have to think, I think the whole question we had at the very beginning of the discussion on combination therapies, this is a very complex disease. I don't think we're going to have a one bullet kills all. I think we're going to have to find ways to combine those things together that are going to make them more effective. But I don't know. I think in our preclinical models, at least, we've had great successes by having staged injections and without lymphodepletion. And there's a big discussion in the field about that as well. So I think we have so many challenges ahead of us. I think that we're just at the beginning. But if we can find a way to provide those therapies in a way that is less toxic to patients, or less, and that provides some type of sustained benefit, whether it is through the cells or through the bystander effect, if you want, I think that's what we should strive for rather than.
And also, back to your question about the cell dose, I personally think there's a threshold of cell dose, but after that, this is not the typical, biopharmaceutical drug development where you have a very predictable PK/PD modeling. A lot of times, even if there's a predictive PK/PD modeling, that doesn't work with the efficacy or the outcome. The cell expansion, all the sense that exists in the CD19 field is dependent on the tumor burden as well. So now you're thinking not just the dose itself, but the functionality of the T cells, the composition of the cell product, and then the antigen load, the burden that will allow the T cell expansion, and then adding to that is the mystery of lymphodepletion, which is crucial for this field. We have been working with NCI trying to come up with the right lymphodepleting chemo regimen, and we're also looking at changes in the homeostatic cytokines such as IL-7 or IL-15 that occurs after a lymphodepletion regimen to really come up with the ideal lymphodepletion regimen. But that alone also, significantly, I think, the paper that came out of Dave Maloney's lab is, three log-fold difference in the cell expansion, simply based on whether you have fludarabine that's added on top of the cyclophosphamide. So when you talk about this, the cell dose question, there's a lot of different dimensions that we're going to have to think through, and the approach has to be totally different than the way we think in the pharmaceutical industry.
Tom?
So thought exercise to turn it on its head. So why do T cells proliferate? It's essentially through signal 2, which is provided by the co-stimulatory element in the CAR T cell. So you have CD3 zeta providing the antigen signal and then the co-stimulatory signal, and both of those in a standard CAR are activated in the presence of antigen. Okay, so part of the problem, more tumor, more antigen, more proliferation. So let's scrap that and let's say let's control proliferation differently. Let's control proliferation with a drug. So based on the schedule and dose of administration of the drug, you can control the proliferation of the cells, independent of tumor burden or anything else. And that's what we're doing. That's the product we're taking to pancreatic cancer, hopefully any day now, has that control mechanism in it. And so it's just it's a different way to think about the challenge of achieving and maintaining a dose.
You've all touched on the personalized sort of nature of a treatment with cell therapy. What role do you think characterization is going to have to play, both in vitro in terms of the cells that you're creating, as well as in vivo, to help make this a successful treatment, sort of across the board? Are we moving towards extremely personalized treatments, or are there going to be ways to treat large swaths of people with one type of approach?
I think it's sort of going through the transition phase. I mean, back to the question of, what is the ideal therapy? My personal experience tells me until you have the clinical feedback, you're not going to know what the best in class, or you don't know what you're really going after. So, the company is doing, sizable studies. I mean, and I think every company is doing a lot of translational biomarker analysis to understand, what cells matter and, what are the cytokines that that can provide some predictive elements to the outcome, whether that's a safety or efficacy. I think all those, hopefully in next, 12 months or so, will give some, light into what you should be looking for. I think, in that context, there's a lot of work that needs to be done. And, this personalized, I think you can sort of think about, in a sense, the field right now is more autologous. People are working on allogeneic. I personally think with the autologous, automation and others will carry for some, near midterm future. But eventually, I do believe, Stewart, next to me, that there is enough science that's out there for you to really think through and coming up with a universal cell approach to the immunotherapy. So, Fate Therapeutics working on, we are working on that too. I mean, we've been playing in this field for the past couple of years, and gene editing is one element, but we put more weight in the ability to differentiate the iPS or stem cells into the functioning T cells, which is, one missing link in, what one can do in this space.
Did you want to add on, Stewart?
Yeah, I mean, just very simply, I think we have to try and constrain the variables here.
Yeah.
We've got variable cells going into variable patients with variable indications, and we're expecting a consistent outcome. I mean, it is laughable, but it's unfortunately an expensive joke. So we have to constrain this. Now, will we get to highly patient-specific therapies? I think it's possible, and to David's point, I mean, there are approaches for that with automation and to constrain some of the variables in manufacture and allow more rapid production for those individuals. But if we look at the broader industry, pharmaceutical industry, our industry is geared toward providing one type of product for many patients.
Yep.
And so I think we have to constrain the product variables and understand and embrace the patient variation as best we can. But again, to David's point, we will only know that from the clinical studies, which are emerging right now from groups like the rest of the team here, and we're working in transplant medicine to understand that better as well.
Excellent.
So can I just? Yeah. I just want to take a counter position to Stewart, which is so all of this variability is a problem when you have a product that you give to the patient, and then it's at the mercy of that variability, right, and the variability in the product itself. If you can control that cell, if you can control its proliferation, if you can control its survival, if you can get rid of it, if it's putting the patient in the ICU, okay, then you have the ability, using that control mechanism, the physician has the ability to adapt the treatment to suit each patient. So it's the same product, but it's adaptable from patient to patient. And the goal of doing that is to make a given product candidate available to a broader set of patients. That's exactly what we're doing in transplantation. Just saying, okay, you don't have a matched donor, fine. We'll use your parent as the donor, and we will manage the safety issue that comes with that by controlling the cells. And if they cause graft versus host disease, we can get rid of them. So I would argue that controllability and is something that I think you see everyone. I mean, Kite's working with cell design labs and others, right, have recognized the value of controllability in making these products potentially more useful and more broadly useful and managing that therapeutic window. So I think that's the answer, or at least a big part of the answer.
It really comes down to what's possible now and what could be possible in the future.
It's also a question of pushing the gas pedal and the brake at the same time. Can we try to do something that get rid of all those things and make it work better?
All right, now, I'm sorry we probably won't get to any more questions from the audience because we're going to need to wrap up, but I'd like to give our wonderful panel one more opportunity to share any closing thoughts. Maybe we'll start with you, Stewart, and we'll sort of go across the panel. Any other key points you'd like to make before we wrap up this morning?
I think that's sort of a cautionary and a positive cautionary note. I mean, so we develop cell therapies and cell-based immuno-oncology therapies. Don't underestimate the complexity of the cells and the patients, but also don't be daunted by it. I mean, we're at a point now where we truly are, we do have I think, sufficient understanding to work with some of the diversity and some of the variability and actually produce consistent product. So, no, let's go forward with the clinical data and make better cells for better therapies. Thank you,
Stewart.
I mean, I think this is a great time to be in cell therapy. I mean, we deal with a lot of uncertainties. There are a lot of questions. But, I think directionally, this entire field is, stepwise. I mean, just think about what the field was like 2 years ago and now. And, that within 2 years, there have been tremendous, changes. And I think this kind of acceleration will continue on, once the critical mass has been created. And I would say, what happens to the cell therapy will very much depend on the people who are in this room.
Thank you, David.
I just echo what David said. I mean, think about it. Patients are being cured today who had no chance previously. They had no option. They're not just getting a few months extra, but they're actually have a chance of being cured of their underlying disease, whether it's a hematologic malignancy or a orphan genetic disorder, et cetera. And that's it's an exciting time because the technology and the science is extraordinary, but also the clinical results that are being delivered today are really quite stunning.
Thank you, Tom.
I think you've heard all the challenges that we're facing, and we've been there for 10 years, and that may push you off. It's actually challenges that drive us. It's our ability to actually overcome those things. And we're not even at chapter 1. We're still in the introduction of the cell therapy space. And there's so many opportunities that I think all this room can actually create new companies and address those opportunities. It's just, really the sky is the limit for this.
Thank you, Christian, and thank you all for joining us this morning.